It's a pleasure to welcome my colleague, Dr. Ray Roos, back to Brooklyn, from whence he came. Downstate helped influence his career in neurology, I believe. He received Downstate's Engel Fellowship that helped support his summer clinical fellowship at the National Hospital for Neurology and Neurosurgery in London. And one of our faculty, Stanley Aronson, a neuropathologist well known to me as well, encouraged him to join the laboratory of the future Nobel laureate Carlton Gadjicek at the NIH. After training in neurology, neurovirology, and neuroimmunology at Johns Hopkins, Dr. Roos joined the Department of Neurology at the University of Chicago in 1996, where he spent most of his career. He rose through the ranks, chairing its Department of Neurology for 8 years until 2004, and currently serves on the faculty as Associate Chair for Education in Neurology and as the Marjorie and Robert E. Strauss Professor in Neurologic Science. His neurologic program has focused largely on neurodegenerative disease such as ALS. He has a distinguished career, publishing over 250 papers, editing books. He's been on editorial boards and on numerous committees. He's also served on several NIH grant review committees, and we actually served together on one of these NIH study sections many years ago at the NINDS. Today, Dr. Roos is going to speak on what, in my view, is arguably the most exciting story in all of medicine. It's a story which I have loved telling my children, my second-year medical students, even my grandchildren. It's a story that has continued to evolve, and one could argue that we still don't know the ending. So it's a special treat for me to hear Dr. Ray Roos speak today on the path from New Guinea cannibalism to mad cow disease to all neurodegenerative disease. Ray.
Thanks, Sue, for the introduction. I have to thank the organizers of the meeting for inviting me to speak about this subject, which, as Sue has mentioned, has had an enormous impact on science and medicine, and a subject that I have been privileged to be involved in for 50 years. I couldn't have done this without the support of my wife, Annette, sitting in the front row there, and I thank her for that support over the 50 years. My disclosures are on that first slide below. As Sue mentioned, an important person and mentor for me was Stanley Aronson, and some of my classmates remember him as a neuropathologist who delivered lectures a bit like one of the Barrymores playing Hamlet. It was very Shakespearean and inspiring. He did many things for me as a mentor. One was that he allowed me to be a work-study student working in the blood bank and living for room and board at Kingston House, an old communicable disease hospital. Also, as Sue mentioned, I had the opportunity to spend a summer at the National Hospital in Queen Square in London, and he encouraged me to do so. I have to say that I could not have done this without an alumni fellowship, so I thank Downstate's Alumni Association for being so important in my career development. The last thing, or perhaps the most important aspect of his mentorship, was to encourage me to apply for a fellowship at the National Institutes of Neurological Diseases and Stroke under Carlton Gadjicek, who was a bit like Raiders of the Lost Ark figure, paramount. I interviewed with Carlton during my fourth year of medical school and was invited to participate and join his lab for a couple of years after that. Carlton was so let me start in the beginning of the story here when Carlton was working in the Walter Reed Army Hospital. He was trained in pediatric infectious disease. He was on kind of a sabbatical in Australia and heard about this unusual disease, Kure. And this is one of his first letters, March 1957, sent to Joe Smardell back at Walter Reed. I'm in one of the most remote, recently opened regions of New Guinea in the Eastern Highlands, in the center of tribal groups of cannibals only contacted in the last 10 years and controlled for 5 years, still spearing each other as of a few days ago and cooking and feeding the children the body of a Kure case. So I have to pause here for a minute to mention that the tribes people, the Kure tribes people in this area of New Guinea practiced a very unusual mourning ritual, which was endo-cannibalism. They ate their own, so that when a Kure victim died, they ate that victim. And the central nervous system tissue, which had the highest titer of the transmissible agent, was given to females and children of both sexes, essentially fitting in with the epidemiology of this disease. It was the most common cause of death among the Kure tribes people, the women and the children. It is so astonishing an illness that clinical description can only be read with skepticism, and I was highly skeptical until 2 days ago when I arrived and began to see the cases on every side. I tell you, Joe, this is no wild goose chase, but a really big thing. Everything in my medical training makes me confident. This class of illness is not my specialty, but I can handle research on it as well as anyone. And as Sue said, he won the Nobel Prize later on, so he was self-assessing himself quite well. So the next clue, interestingly, came from a veterinarian pathologist. And I have to mention that Gadashek obtained central nervous system tissue from Kure victims, inoculated it into rodents, kept them for weeks, maybe a month or two. Everything was unsuccessful in transmitting this disease. And the veterinarian pathologist was Bill Hadlow, who had seen an exhibit about Kure and wrote this letter to the editor of Lancet in 1959. I'd like to present a few brief comments on the similarity of two progressive degenerative disorders of the CNS, namely scrapie affecting sheep. And this is the sheep you see scraping the pole here, and Kure affecting the Kure natives in the Eastern Highlands of New Guinea. I do not suggest that these diseases are identical or even counterparts, but in my opinion, their overall resemblance is too impressive to be ignored. And on the left part of the screen here is a vacuolated neuron from Kure, and next to it is a vacuolated neuron from scrapie. So Hadlow went on to tell Carlton what to do. Despite the lack of indications suggesting that scrapie is an infectious disease, of course there was no inflammation, this disorder can be induced experimentally, that is transmitted in the sheep and in the closely related goat with incubation periods of years. Thus it might be profitable, in view of veterinarian experience with scrapie, to examine the possibility of the experimental induction of Kure in a laboratory primate, for one might surmise that the pathogenetic mechanisms involved in scrapie, however unusual they may be, are unlikely to be unique in the province of animal pathology. And Carlton kept this in mind. Scrapie was known to be somewhat species-specific with respect to transmission. So Carlton took Kure central nervous system, inoculated it in the central nervous system of non-human primates, and in 1967 transmitted Kure to non-human primates. And the vacuolated neuron became this rather remarkable status spongiosis in the brain of this non-human primate. So the next question came up, what other diseases are transmittable like Kure? And very early, when Gattashek was in the Kure area. He sent central nervous system tissue obtained at autopsy to the NIH to Igor Klatso, and Igor wrote back as early as 1957, Kure seems to be definitely a new condition, without anything similar described in the literature. The closest condition I can think is that described by Jakob and Creutzfeldt. The etiology of Jakob-Creutzfeldt condition is entirely unknown. Only about 20 cases have been reported. So he was a real scholar. This was a very rare disease, little known, and Klatso was able to realize that these were similar. And with that in mind, Gattashek obtained Creutzfeldt-Jakob central nervous system tissue from neuropathologists worldwide. And in 1968, the year I graduated, Downstate transmitted Creutzfeldt-Jakob into non-human primates. I had the opportunity really to describe these cases clinically because a large number, by the time I arrived in the lab, had been collected and transmitted. And later on, in 1976, Gattashek received the Nobel Prize for the transmission of subacute spongiform encephalopathies, as these diseases were called. So what are these diseases? Kure, Creutzfeldt-Jakob, and maybe some other very rare ones. Interestingly, they have similar non-inflammatory clinical syndromes and pathology. No inflammatory diseases here. And they're transmissible by an unconventional agent. Long incubation period. So Gattashek, between the inoculation of a non-human primate to transmission of Kure, was 18 months. And in humans, for a tribespeople, it's been documented that transmission of Kure, human to human, from an endocannibalism to transmission of Kure, is over 3 decades. The transmissible agent is resistant to physical and chemical inactivation that nucleic acid-containing agents are usually susceptible to: heat, exirradiation, formalin, raising public health issues. And in fact, you could take a microscope slide of a section of Creutzfeldt-Jakob that's been formalin fixed, heated, scrape that, and transmit that tissue to an appropriate host. I had the opportunity really to do the first epidemiological study of Creutzfeldt-Jakob, and we looked at a number of the sporadic cases, wondering whether there was some clue with respect to transmission. And in fact, this was basically a pretty negative epidemiological study. Subsequently, unfortunately, iatrogenic Creutzfeldt became apparent, and there are dangers, or were dangers, of transmitting disease from blood transfusions, corneal transplants, growth hormone injections, dural transplants, and public health issues emerged, and appropriate strategies have been taken up. One of the interesting aspects of the spongiform encephalopathies is some of these diseases are familial. And we published the first positive transmission of these inherited Creutzfeldt-Jakob, and then we're involved in a very unusual spongiform encephalopathy, fatal familial insomnia, in which patients, part of their autonomic abnormality, are unable to sleep, and subsequently are involved in transmission of this disease. So what's the nature of the transmissible agent that has such long incubation periods that can transmit familial disease? And so a second Nobel Prize was won in this group of diseases by a neurologist, Dan Prusna, who found that the transmissible agent was a normal protein, same amino acid composition as that normal protein, but that this protein, this prion protein scrapie form, was misfolded, a misfolded form of the normal cellular prion protein. And the prion hypothesis, which is an important one, is that this scrapey, misfolded prion form acts as a template. And you see here in blue that abnormal form that takes the normal cellular orange form and converts it to a misfolded form, which accumulates, kind of like an infectious agent. It's kind of like replication. And what we know now is that in the inherited cases, the mutated PRP, because in this case there's an amino acid abnormality, is more likely to adapt the misfolded conformation. So these diseases were, although important from a public health perspective, were a little bit of a curiosity up until the mad cow epidemic, bovine spongiform encephalopathy epidemic. And you see here cases over the years in the UK of bovine spongiform encephalopathy, thousands of cases, but this really is just a shadow of the real figure, which was very impressive. There were two important questions that came up when the bovine spongiform encephalopathy epidemic occurred. And the first one had to do with its origins. How did it appear? And interestingly, what we think happened is that, well, we know that remnants of slaughtered cattle were recycled back into cattle feed, kind of like the endocannibalism of Kure. And some of this slaughtered cattle presumably had prion contamination, maybe from some scrapie, perhaps. And second, that the rendering process, a meat processing operation, had been changed to save money, allowing a lower temperature and therefore sufficiently high prion levels for efficient transmission. And the second big question had to do with concerns about transmission to humans. And the UK Minister of Health in 1995, with his daughter eating a wimpy hamburger, said there's no conceivable risk of BSE, bovine spongiform encephalopathy, being transmitted from cows to people. This was December 3rd, 1995. And Bob Will, a neurologist, in March 1997, in the past few weeks, we believe that we may have identified a new clinical pathological phenotype of Creutzfeldt-Jakob, which may be unique to the United Kingdom. This raises the possibility of a causative link between BSE and Creutzfeldt-Jakob. And in fact, here's in yellow the BSE, and in red, variant Creutzfeldt-Jakob disease. And it's clear that there was a link between these two diseases. Fortunately, the worries about Creutzfeldt-Jakob calmed down a bit, and there's probably been a couple of 100 cases in the UK, elsewhere in Europe, and partly because of measures taken in order to change slaughterhouse procedures, slaughtering cattle that might have been infected. I became very concerned about this, being a member of the FDA committee on spongiform encephalopathies, and the question came up, is there mad cow disease in the US? Somebody sent me this cartoon. It says, except at branding time. Fortunately, we've had no mad cow in the US, but in fact, spongiform encephalopathies are present, animal spongiform encephalopathies. This is an old slide from 47 to 1977, scrapie outbreaks over the U.S., and you'll be concerned to know that when the farmers found scrapie in the herd, they would sell meat on the open market. And we still have scrapie in the U.S. and really worldwide. And not only scrapie, but in North America, we have chronic wasting disease, a prion disease of free-ranging deer and farmed elk, known for almost 50 years, and outbreaks of CWD are shown in North America. This is from 2017, and this disease has gone from the west to the Midwest and actually to the East Coast. So what's next in spongiform encephalopathies and prion disease? Of concern was whether misfolded proteins, which are key to prion disease, are present. We know they're present in neurodegenerative diseases. Are they important in the pathogenesis of these diseases? So just so everybody's on the same base with respect to neurodegenerative diseases, these are the diseases we don't really know the cause. They're not inflammatory. They're not a result of ischemia. They're progressive. Usually there's an inherited and sporadic form. And what makes them neurodegenerative diseases is they affect one or more system of neurons. And they're associated with misfolded and aggregated proteins. And you see here a number of neurodegenerative diseases, ALS, and we have a number of misfolded proteins in yellow, Parkinson's disease, Huntington's disease, Alzheimer's disease. They're all misfolded aggregated proteins that are present in these neurodegenerative diseases. And the question came up, are misfolded proteins pathogenic in neurodegenerative diseases, such as ALS, which is a disease that I was studying really for other reasons, but this misfolded protein theme kind of came back to hit me. And the concern was, are any of these diseases transmissible? And that may be the case rarely, not so clear. But more important, is the prion hypothesis important in the spread of these diseases in an individual, in a central nervous system, in which misfolded proteins are templating a normal protein and therefore causing the disease to spread. So let me talk a little bit about ALS. Like a lot of our neurological diseases, they're very bad ones, in this case progressive paralysis leading to death. What's opened a little bit of a window on ALS is understanding its genetics and that there are mutations in SOD1 and C9orf72. And there's a transgenic mouse in which mutant SOD1 is present that develops ALS and serves as a disease model. So what's the normal cellular response to misfolded or unfolded proteins? And can one harness it to treat ALS or, in a way, all of neurodegenerative diseases, kind of like a generic approach to misfolded proteins? So a busy slide, but there are important pathways, especially this PERK pathway of the unfolded protein response, which phosphorylates this protein, EIF2 alpha, which is key to translation, conventional translation. When this gets phosphorylated, translation of proteins is inhibited. There are fewer misfolded proteins. But what happens is when one excessively makes misfolded proteins, then an apoptotic cell death pathway diverts the UPR, and it's overwhelmed. So we used a genetic approach to take this mutant SOD1 transgenic mouse and have it haploinsufficient for PERK, and had a very shortened lifespan of this mutant SOD1 mouse. And we were able to enhance the UPR by having a mutant SOD1 mouse that had a mutation in GAD34 with a markedly longer survival. So by perturbing the UPR, you could manipulate this genetic ALS mouse rather prominently. And the question was, could we have reagents in humans? Because these were genetic approaches. And what kinds of reagents could we use? And we now have a few reagents that target PERK, GAD34, and this apoptotic protein CHOP. They knock down these particular messenger RNAs, and we've begun to look at them. For example, this is a GAD34 shRNA that knocks down GAD34. Here's the normal mutant SOD1 survival controls, and this mouse that's treated with this viral vector-delivered GAD34 shRNA has about 40 days prolonged survival. We're continuing to investigate these reagents. We've also looked at the UPR C9orf72. So this is an important mutation. It is the most common cause of familial ALS, frontotemporal dementia, and a very large group, 5 to 10% of sporadic ALS. And this mutation is very unusual. It takes a repeat here in a non-coding region. But this repeat is unconventionally translated into what we call DPRs that are toxic. We were able to express one of these DPRs in cells, and you see here phosphorylated EIF2 alpha in these cells. So what we think is happening is that this repeat-associated, unusual, unconventional translation makes DPRs which aggregate, turns on the UPR, phosphorylates EIF2 alpha, and the only translation into proteins is an unconventional one. So we have a pathogenic self-sustaining cycle. And we're interested in models of this disease using our reagents. And then, not so long ago, and this is one of 3 papers on Scrapie, not from my lab, that showed that the UPR and manipulation of the UPR can be neuroprotective in Scrapie. So I had the funny feeling that here I have been running around for 50 years, and I'm kind of now taking a look at these prion-infected mice with the reagents I have. And then I thought, my God, I'm back to Downstate after 50 years. So in a way, I'm back at the same place and really very happy to be here. So I've learned a lot from these diseases. Non-inflammatory diseases and inherited ones can be transmissible. Unconventional agents with unique properties can be active not only in neurological diseases. Diseases of animals and ones found in exotic areas can provide clues that change our thinking regarding medicine and science, and misfolded proteins are important targets for therapy of neurodegenerative diseases. And here's just a list of some of the people that have really done the work and helped me, and I'm happy to take questions. Thanks. Yes.
So, first of all, that was a wonderful talk. It's always nice to learn new diseases and new physiology, which most people don't learn today. So, thank you. You did a great job. I have one perhaps naive question. I understood the concept of the misfolding of the prion protein to result in disease. But before you can have a misfolded protein, you have to have a protein. So what is the implication or the consequence of having properly folded prion protein in your brain?
So I guess the question is Larry, right?
Yes. That's not the question, that's the speaker.
The question, in a way, has to do with what's the normal function of the cellular protein. And I don't think that's exactly known, although very well investigated, and people have made knockout mice of varying sorts. I'm not sure that that answer is exactly known. I might just mention that besides PRP, there are a number of normal proteins that get misfolded, like prion protein, and some of them are critically important with respect to functioning, for example, with respect to memory. So these can be important proteins, and I have no question that PRP is important. It's even been implicated in Alzheimer's as well. But I think the jury's still out with respect to its function. Yes, Fred?
Are the epidemiological studies that trace back from the mad cow disease issue, trace back to where the cattle came from, what farms, and is there any sort of a pattern of these, obviously, disease-causing proteins being transmitted because they've been moved around from place to place?
It's a good question, and I have no question that that's been really well looked at. In a way, some people think that there was some scrapey sheep that got contaminated the feed. Another really has to do with the theory of what causes sporadic Creutzfeldt-Jakob disease. And it has been hypothesized that there's a stochastic misfolding of a PRP in an individual who's destined to get sporadic Creutzfeldt-Jakob. And this acts as a template increasing the spread of this misfolded protein. And so kind of an answer to your question is, in the case of BSE, maybe a cattle rarely has a cow rarely has a stochastic change in its PRP and developed a prion disease in that cow. And as I said, this putting back into the feed the dead cow remains is a dangerous issue. So the answer is, again, we don't know for sure, just hypotheses. Yes?
In the case of the non-mutant prion, it would occur to me that kind of an event might happen very frequently, and that prion diseases ought to be quite common, and yet they don't seem to be. So is the event, is the particular type of misfolding very, very rare if you were to try to predict how often it is, for example, in the laboratory?
There were some very interesting studies taking appendices in the UK. Those tissues were available, and they've done a number of studies looking at the appendices because they wanted to know how prevalent variant Creutzfeldt might be in the general UK population. And they did this, and there was a certain incidence, and which was a little bit higher than I thought. And then they took appendices before the mad cow epidemic, and they actually found a somewhat similar number, in a way. And interestingly, all the variant Creutzfeldts have had a particular polymorphism in the PRP. One amino acid is a methionine or a valine, and all of them have a specific met-val or met-met or val-val. These appendices turn out to be much more mixed with respect to the polymorphism. So what does that mean? One of the questions is, could you have infection without clinical disease? So we don't know, but it could be that, yes, there are changes in people at time without overt clinical disease. Yes, Sue?
Or, alternatively, it may be that only the homozygous population already have the disease, and now we may have a second.
A second wave is a concern here, right. All of these variant Creutzfeldt-Jacobs, and basically there have been no cases over the last couple of years, have had one polymorphism, and maybe there's going to be a second wave. Yes?
Obviously, the protein is manipulable by various molecular mechanisms. Why is it so resistant to destruction? If you can manipulate some of the channels, so to speak, why is it such a different thing that it doesn't respond to formalin, doesn't respond to heat? What is it that makes this almost non-biological?
I think I need a biophysics background to answer that appropriately, but I think this aggregates and gets very tight, and it's protease resistant. It's not, you can, in fact, dissolve it, but it's certainly much more resistant than a normal protein. And I think it has to do with its aggregation. And people have structural studies of PRPC as well as PRPH. And no question that there's a very dramatic change in its structure from a crystal perspective. Yeah.
Just a question. So why these proteins target the brain? Why not go into your liver and cause liver?
That's a really really good question. So and maybe I should have made mention of this. So why neurons? Well, neurons are very special because they're non-mitotic, non-proliferating cells. So they have the opportunity to build up these misfolded proteins. Remember that are templating. And so perhaps over time, over the years, these fill up with this pathogenic toxic material. But it turns out that people have implicated prion disease also in non-neurological diseases like even diabetes, and misfolded proteins might be very important in all of these. But why the CNS so specifically? And I think it has to do with the nature of a neuron. Yeah.
So, as a gastroenterologist, I spend a lot of my time in the colon. And
I'm sorry about that.
I have to go back to my mother. So, nonetheless, I may be slightly overstating this, but if you biopsy the right side of the colon in patients, sometimes you find you can stain for alpha-synuclein. And these are the patients that will go on to develop Parkinson's disease in the typical way that we recognize it. Taking off on High's point then, can you see any other organ involvement prior to brain involvement that might presuppose the disease actually starts somewhere else? Like when you swallow the cannibalized material, it enters your GI tract first. Is there anything that happens in the GI tract, whether it's microbiome related or not, that then determines whether that protein will be able to be manipulated or discarded to cause the disease or not?
So really it has to do, your question, with tropism of this pathogenic agent, which partly depends on the route of transmission. For example, if you inoculate intracerebrally, it's a bit different than if you orally transmit this disease. And extensive studies have been done looking at every tissue over time by Gadashek and others. And it goes in the GI, into lymphatic tissues, into muscle, into the central nervous system. And that's part of the danger in from a public health perspective, no question. And some of the prion diseases have slightly different tropism. For example, variant Creutzfeldt-Jakob is especially lymphotrophic. So complicated answer, but it's certainly in other tissues. Yes?
I just want to focus a little bit on ALS. You know, there
been very few people who had long-term survival. I wonder if you have ever had access to such patients, and if not, is there a way to get access to do studies on these patients to see what is it that's allowing them to survive so long?
Yeah, I've never seen anyone with ALS that survived the disease and overcame it. We do have some individuals who have a longer lifespan, perhaps 10 or more years. In fact, it's said that 10% can live 10 or more years, especially some of the inherited cases, that's true. So it's probably very much a syndrome in that respect, no question. And so maybe it's a slightly different disease, especially in some of these inherited forms. Yes?
I'm wondering whether or not you've seen a relationship, or do you think there could be a relationship with diseases like multiple sclerosis or neuromyelitis optica or some of those diseases with the Alzheimer's and ALS and so forth based on the disease of the changes?
Well, MS and MO are very inflammatory diseases, and so they're certainly different than what we call neurodegenerative disease processes. There may be some aspects to those diseases that involve misfolded proteins, but perhaps they're a little bit distinguished from neurodegenerative processes.
I have a comment, and I guess we can move on to the next speaker, but there's a very fun book that came out in the 90s called Deadly Feasts for people who want to read a light reading history of prion disease epidemiology. I don't know if you endorse it or...
Absolutely,
or
yeah.
But it's a very fun history of prion disease. It's called Deadly Feasts.
Right. And there's another one by Carlton Gadgetek, Notes from the Field.
Yes.
Fascinating.
Thanks very much.